Hello, everyone, and welcome. We want to thank those intrepid souls who actually crossed over to Radcliffe today, despite the weather. Winter is fully upon us. Thank you very much. I want to welcome you to our Science Lecture Series. My name is Sean O'Donnell. I'm the Associate Director of Academic Ventures, and I help to oversee the lecture series. I have the opportunity and privilege of working with Immaculata DeVivo and Alyssa Goodman, our science faculty directors, whose vision has really helped launch a year-long exploration of this idea of what is the undiscovered in science. There's clearly so much more for us to know, but in practice, of course, for scientists, that often means that they are setting off to find one thing, and what happens when they find something completely different. This notion of serendipity is not just romantic, but it's also a very practical experience, and we are really honored today to have Professor Gautam Dantas with us today, whose story about his own surprising and unexpected discovery really redirected his research and led to a whole decade worth of a new research program. I will let Professor Dantas tell that story himself, of course, but I do want to briefly introduce him. And I'd like to do so, if you don't mind, by reading his Twitter handle. He is a bow tie enthusiast. He is a husband, a father, microbial ecologist, genomicist, computational biologist. He's also a home brewer, gardener, foodie, and pretentious cocktail mixologist. I hope he's serving up some wonderful cocktails for us today in his stories, and should also point out perhaps that he is at the same time the professor of pathology and immunology and biomedical engineering and molecular microbiology at Washington University School of Medicine in St. Louis. And he is no stranger to Harvard. He was here in George Church's lab, and he met with our students today, and he clearly has a desire for mentoring students and leading them and directing them in this process of becoming scientists and inquisitive minds. And with that, I hope that you will join me in welcoming Professor Dantas to the stage today. Thank you.
Well, thank you, Sean, for that very kind introduction, and thank you to the Radcliffe Institute for this opportunity to come back to an old home. Before I get started, I want to say something that I personally believe and I think reflects the opinion of most of my good colleagues, and that is all good science is team science. And if there's anything of importance that I say today, it's because of these fine folks that I have the privilege of working with. I'm really here as their spokesperson. Before I get to the science aspects, I do want to mention this role of serendipity in everything that you'll hear today. So I was a postdoc in George Church's lab from 2006 to 2009, and I came here to ostensibly study microbial engineering for biofuels, right? This idea that we could convert plant sustainable matter into replacements for petroleum. And the approach that myself and this grad student, Morton Summer, took were to try to enrich microbes from the soil that could survive on plant-based toxins as their sole source of carbon, with the hope of understanding that genetic machinery. And we thought we'd be good scientists and set up an appropriate negative control. And luckily, our naivete led us to this idea that, okay, the type of compounds that no microbe should be able to consume as a sole carbon source would be antibiotics. So we went to off-the-shelf drugs that we could find in George's lab, set up the experiments, and lo and behold, a week later, we got very poor results on the plant-based toxins, but we had gangbuster growth on all of the antimicrobials. We were surprised. Most reasonable advisors would have said, that's great, why don't you focus on what you're doing? George is wonderfully not that type. He got as excited as we did and said, stop the biofuels work for a while and figure this out. And the rest is history. Basically, everything I'll tell you about today, every part of our interest in antimicrobials, even the ones that'll seem like they were designed from scratch with this prescience a priori, really was because we decided to follow up on that slightly weird experiment. So let's step back for a second and think about what we're actually going to discuss, and that is antimicrobials or antibiotics. So the definition of an antibiotic, just to get sort of the language straight, is any chemical that it either inhibits the growth of or kills microbes. And today I'm going to exclusively be talking about the group of antimicrobials that are antibacterials, these compounds that can be used to kill bacteria. One thing that you should note is what they're not is they're not antivirals, right? And so this is a CDC warning that very clearly tells you, and you really should heed this, that anytime you have a viral infection, you certainly will be doing no good if you take antibiotics for those viral infections, and I'll show you pretty soon that you're probably doing something bad. So one thing to recognize as to why these particular drugs work is because they target the most key and conserved processes of bacterial life. They destroy the bacteria's ability to have a cell wall, so their guts bleed out if they are impacted by things like penicillins, or they gunk up the ability for them to replicate. And the reason I bring this up, rather than really to discuss molecular mechanism, is to mention that Paul Ehrlich called antibiotics magic bullets. But we really should be thinking of them more as magic shotguns or magic nuclear bombs, because every bacterium has this target. So when you ingest an antibiotic, thinking that you're going to be targeting a specific pathogen, you're actually impacting every single microbe in that ecosystem, for instance, in your gut. And so we realize that even though it might be warranted use of this really important chemical, you're going to have potential collateral damage. And it's through that lens that really I'm going to be describing work that people in my lab have done to understand what that collateral damage might be due to both warranted and unwarranted use of antimicrobials. One thing that we have observed in that particular vein, especially by looking at retrospective studies, is any time that antibiotics are used in, for instance, the human population, that those antibiotics do have impacts on all of the good bugs as well as the bad bugs. So down here, you look at this particular plot, and you can observe that virtually at any point in life, at any time point, you'll notice that there are some kind of malady or some sort of problem that occurs on the human side, even if the antimicrobial was warranted. Now, perhaps the greatest response that a microbe might have to trying to be killed by an antimicrobial is to respond to become resistant to it. And this idea of antibiotic resistance is something you'll see as a common feature that I'll discuss through the rest of this lecture. And it turns out the history of antimicrobial use in the human population is intricately linked with antibiotic resistance. So we're here to talk about serendipity. The whole field of antimicrobials is really thanks to a serendipitous accident at the hands of Sir Fleming, who happened to have one of the greatest opportunities to win a Nobel Prize because he got a little bit sloppy when he went on vacation, right? So he left out these plates that we all have heard about, found the penicillium mold, wiping out this potential pathogen. But the interesting part for this is that this natural product antimicrobial penicillin, first discovered by Fleming in the 1920s. The first actual treatment, because of the amount of development it took, was about in the 1940s. But the first reports of resistance to this compound predated that by over a decade. And this is something I'll keep coming back to, is that antimicrobial resistance, even though we hear about it in these scary stories in the media as being associated with the clinic, is actually a natural feature of all microbes. You also take the example of the sulfonamides discovered by this German chemist, Gerhard Domach. The sulfonamides were a synthetic class of compounds. Their first treatment was in the mid-30s, and this is a little bit more intuitive that the first reports of resistance were about 4 or 5 years later, though we've discovered that that was, again, an underestimate. Resistance to those particular compounds was well before humans came on the scene. And so this particular plot shows you the sort of zoomed-out view for every class of antimicrobials. So what you're seeing on the x-axis is time. You're seeing along the particular plot every single important class of antimicrobials used in the clinic. And when blue shades to red is the very first reports of clinical resistance. Another way of interpreting what you're seeing on this particular plot is antibiotic resistance in pathogens is not a question of if, it's just a matter of when. Death, taxes, antibiotic resistance, right? All of them are predictably going to come up very, very fast. And this encodes an incredible burden on the human population. Now, some of these numbers have been challenged, so I'm not going to focus too much on the specifics, but we do know that a large number of people, almost 1 million people around the planet right now, die due to drug-resistant infections. And This might scale up if things don't change by about 2050, based on this UK Prime Minister's report from a few years ago, to maybe one person dying from a drug-resistant infection every 3 seconds on the planet. Now, of course, the human life is the most important in this equation, but it also costs a lot of money. So the US economy is estimated to lose about $55 to $60 billion per year because of the treatment of drug-resistant infections. And if that 10 million number is to be believed, it also scales to about a cumulative hit to the global economy by 2050 of $100 trillion. So $100 trillion, for those of you like me who don't have access to that many zeros, is about 6 years of the US GDP wiped out. And I will warn you, I'm a little bit sorry that this is going to be a slightly bad news bears talk, that exactly at this time, when we've got this massive increase in drug-resistant infections, exactly when we need new drugs to be coming to market, we don't. There's lots of reasons for this. So what we've decided to do as a group is focus not only on trying to understand how we might come up with new antimicrobials, but take the parallel complementary view of saying, if we were to discover key features about antimicrobial resistance that we might be able to counteract, that could be part of this particular pie. Now, this gets a little bit molecular in these details, and I'll try to keep this at the level that is still relevant to this discussion, and that is to ask, now that we know that resistance is a problem, how is it that bacteria evolve resistance, right? When they see these antimicrobials, how do they make changes in their genomes to become resistant to these compounds? And they do it in the way that bacteria evolve any property. And it's in one of two ways. One way is the way that we evolve. That is, we pass on our genes over to our progeny. Bacteria do this by dividing. And then mistakes occur during those replication processes. And if a particular mistake, a genetic mutation, happens to give your progeny a selective advantage, it's going to now take over the population. And say that selection happens to be antibiotic resistant, now that particular resistant mutant, that resistant kid, if you will, in the bacterial population, will take over. That's called vertical transfer. But bacteria and archaea, the other two domains of life, can do something that's much more spectacular, something that, unfortunately, so far, we can't do as humans, and that is, in one fell swoop, exchange large chunks of DNA, the genetic material, with their neighbors. So they don't have to wait until they have kids. They can pass on traits to other members in their population. And as it turns out, if those particular pieces of DNA encode antibiotic resistance, in very short timescales, you can take an entirely susceptible population of microbes and convert them to be multi-drug resistant. And analyses that many groups have done now suggest and show that because of the huge selection pressure of surviving under antibiotic stress, microbes then use this particular method, this idea of horizontal gene transfer as opposed to vertical transfer, to become antimicrobial resistant. And so what I'm going to talk about are a few stories that we've used modern genomic methods and computational methods to understand the burden of horizontal gene transfer in terms of who's the source of these resistance genes and how do they eventually end up in the clinic to cause the type of problems I just talked about. Now, one thing that we as biomedical scientists have learned over the last, say, 10 or 15 years is if you're a scientist worth your salt, you need an ohm at the end of what you study. The genome, the epigenome. Not to be left behind. We at the group that study antibiotic resistance were fortunately given the resistome, actually a very elegant concept put forward by Jerry Wright from McMaster University about 12 years or so ago, to describe all of the genetic material in any arbitrary microbe that will allow you to be resistant to a particular antibiotic. So how do you study the resistome? How do you study all of these resistance genes? Well, you could certainly turn to the foundation of microbiology, this idea of domesticating the microbes by bringing them into the lab. And we should certainly be happy that this occurs because if a clinical microbiologist wanted to diagnose your infectious agent, this is what they would do. And in the modern era of genomics, you could then sequence the genome of that organism, infer the resistance property. The problem, though, is we've really realized over maybe the last 2 decades that most microbes live in microbial ecosystems all over, in the soil, in our bodies, virtually anywhere we look, and most of those have not been cultured. So how do you study things that have not been cultured, that we've not been able to domesticate? Well, we can turn to sequencing. This idea of now looking at just the DNA, the genetic material in the organisms, and infer what type of functions they might have. In this case, the function of antibiotic resistance. So at the practical level, this is literally just smashing up all of the microbial cells, harvesting their DNA, putting them through a sequencer, and then inferring what the resistance might be. And this might seem like you've got the whole part of the pie. Unfortunately, you're only iterating on the known. So what does that mean? It means that all of our databases of known antibiotic resistance genes have been inferred from that small, tiny minority of bacteria that have been cultured. And so then when you use sequencing methods, you're just relearning the few tiny ones that you've known before. So what we've done is try to complement that part of the cryptic resistome, the part of the pie that is the undiscovered, to try to improve our ability to not only harvest out and bioprospect genes that might be a problem in the clinic, but also to try to be sort of prescient about what might eventually occur in the clinic. So what is this complicated term called functional metagenomics? That is the method that we use. So the method itself was co-developed by Joe Handelsman, who's at Wisconsin, and John Clardy, who's here at Harvard Medical School, back in the late 90s, where they discovered that much like the natural process of horizontal gene transfer, if you could come up with a method that forces that to occur in large scale, then you might be able to use model organisms like E. coli and test their ability to express random chunks of DNA from any organism to encode resistance. And the steps are the following. You extract the DNA, you clone it in to a library in E. coli. This is an easy-to-use bacterium. You get millions of these random chunks of DNA in E. coli, and then you turn back to culture. So in this case, much like a clinical microbiologist would do, you use Petri dishes that have antibiotics in there. The bacteria that grow now have a new chunk of DNA that is a resistance gene, and then you figure out what's going on, right? What are those genes by sequencing? What our lab has basically done is taken that basic engine, married it with sequencing technology, and effectively reduced the cost of doing this by about 1,000-fold. Now, let's say for the sake of argument that you completely zoned out for those last two slides and don't care about any of those technical details. The only thing that you need to take away from what I just said is that with this method, you can start out with any arbitrary group of microbes, whether they're cultured or not, and after pumping them through this particular pipeline, which is now a lot cheaper to use, you'll get a catalog of all of the genetic material that is compatible with horizontal gene transfer to make a particular bug like E. coli resistant to antibiotics. So we apply a method of this type to then interrogate all sorts of different microbial communities for their potential to contribute antibiotic resistance to the clinic. So this is one way in which we view the world, this cartoon schematic of interacting habitats. And the reason I put this up, and you'll see this a couple times, is to recognize partly that microbes don't care about our definitions. Right? We might define something as a habitat boundary. Microbes can traverse those. And so what we really like to understand from an ecological perspective is that even though resistance really is most acutely problematic in the clinic, because microbes in different habitats could themselves transfer over eventually into the clinic, or their genes could transfer over, we would like to understand all of the interactions that are shown here. So what I'll walk through is a couple of habitats that we've particularly focused on to discover resistance genes, to assess their risk of getting into the clinic, and hopefully also to mitigate those particular risks, right? And I'll start out by not going into too much detail and just giving you a couple of highlights that we've published on from our investigations of the so-called commensal microbiome. So these are the good bugs, for the most part, that live in and on our bodies, right? The one that's been studied the best is the gut microbiome. So again, this is the trillions of microbes that live in us. They're basically another organ. And because they're constantly subjected to antibiotic pressure, it makes sense that they might be a source through which pathogens could pick up resistance genes. So one of the things that we were surprised to find in collaboration with Rob Knight's group as well as Maria Gloria Dominguez was that through them we got access to fecal samples and oral samples from an Amazonian tribe at first contact. I can't tell you how do you convince a group of people who have never seen the outside world to give you a fecal sample. I think that's probably how our first alien contact will be. You'll be taking microbiome samples from us. But Erica Pearson, a genetics graduate student in my lab, accessed those samples, used the methods I just described, and we were shocked to find in these people who had no history of any antibiotic use, resistance genes against modern antibiotics. And you'll see when I get to the soil why this makes sense, but we thought this was an important thing to recognize, that resistance against these compounds is not something that we invent through our use. It already exists as a natural feature of these ecosystems. We just give selection pressure by using the antibiotics for them to amplify. Okay, now I'll give you another example of something that's at the other extreme of the spectrum, where we know in vulnerable populations they get a ton of antibiotics. And that is, for instance, in this case, preterm human infants. So we know that rates of prematurity are going up, and especially when we look at very low birth weight infants. So these are infants that are born about 10 weeks too early. These kids are highly immature in terms of their immune systems, and because of their risk of infections, we give them a ton of antibiotics. Well, not me, I'm not a physician, but the field. And so, for instance, in this cohort, which is describing kids born in St. Louis at our children's hospital, virtually 100% of these kids born 10 weeks too early get antibiotics as soon as they're born for the first couple of days of life. And as you see in the plot next to the histogram, the top 2 drugs that they get while they're in the neonatal ICU are antibiotics. 4 of the top 8 drugs that they get are antibiotics. So we step back again from an ecological perspective, and you think about the fact that these kids require this commensal microbiome to set up. What you're doing with each of these insults is you're carpet bombing an ecosystem that should be setting up normally. So we wanted to assess what the collateral damage might be. And so one thing we found, and this was work led by Molly Gibson, a graduate from our computational biology program, in collaboration with a couple of pediatricians at WashU, Barb Warner and Phil Tarr. We had access to a large cohort of fecal samples. They actually have about 75,000 such fecal samples banked, which is probably the largest collection of poop in one particular location. And we accessed only 400 of those in 2 groups of kids, ones that had lots of antibiotic exposure and ones that didn't have very much. And the thing that I just want to emphasize, for the non-clinical microbiology aficionados, the main thing that Molly discovered that was surprising to us was the identity of the bacteria that was surviving all of these insults in these kids. And so, again, don't worry about the specific names, but you'll have to take my word for the fact that the bugs dominating the guts of these very vulnerable kids are the bad guy bugs on the CDC list, right? These are not bacteria that we associate with good gut health. They're the bugs that usually cause nosocomial or hospital-acquired infections. And perhaps now that you think about the selection pressure, it's not all that surprising. Who else can survive those insults except the really hardcore drug-resistant bugs, right? But the silver lining there is that through a series of computational approaches, again, the aficionados call it machine learning. That's partly, because it sounds fancier. It's really pattern recognition using computer algorithms. So what Molly did was she took all of this data that she had gathered in terms of who was there in the microbiome and their resistance genes and trained a model and was fortunately able to predict how the microbiome would change based on just a couple of these features with about 85% accuracy. Why is that something to consider as being important? Because it suggests that a potential future of personalized medicine where each of us, anytime we get sick, we might be able to get our microbiome sequenced. As these type of methods improve, your physicians might be able to take the information about your specific microbiome state and tailor your antimicrobial therapy in a way that not just kills the infection, but protects your microbiome. Right? So we're hoping that this is where this will go. Now, a picture tells 1,000 words, and I can't think of anything besides this particular questionable parenting event in really telling you that resistomes cross habitats, right? And so we decided to take this concept, and there's the sort of slightly more boring version of that slide, where fortunately, through a number of international collaborations, led by Erica, who I already mentioned, and Pablo Tsukuyama, another graduate student in my lab, we accessed samples from a village in El Salvador and a slum outside Lima, Peru, to look at potential interactions between microbes and resistance genes between people, so they are fecal samples, and as many environmental samples we could get at the same time with GPS coordinates, right? So soil samples and sewage samples and subsistence animals and pets. And again, I won't belabor all of the incredible data analysis that these folks did, and just jump to the conclusion that we think has impacts on public health, and that was they were able to identify using this analysis key hotspots for resistance gene exchange. right? So what this means is they identified particular features, particular habitats between these people in the environment that had clear evidence for having the same resistance genes in those habitats in humans as well as the environment. And we found 2 such key habitats, which could be really impactful for public health in El Salvador and in Peru. So across the top panel, you'll see pictures of chicken coops in El Salvador in the village, and that appeared to be one of these hotspots. So this is the droppings from the chickens were subsistence animals, had exactly the same resistance genes as the humans as well as the soil, implicating those chickens as a route moving between humans and the environment. The reason this is important to consider, especially here in the US in terms of its translational impact, is the folks in El Salvador in this village don't use any antimicrobials in rearing their chickens. But almost any chicken that you or I would eat has probably been grown under the pressure of lots of antibiotics. If you think of what's happening here, consider what might be happening when we're eating chicken under antibiotic pressure. The story was a little bit different in Lima in terms of what we found there. There we were able to implicate the sewage treatment system as a hotspot for such exchange, right? So this is a massively crowded ecosystem. All of the effluent sewage goes through a single treatment plant. It gets cleaned up based on pretty sound civil engineering principles. And then once that water is cleaned, it has to be dumped somewhere. Now in Boston, that's dumped in your harbor. Far out, but still in your harbor, right? In Lima, being a desert, that water is used to irrigate every single field and every single place that requires water. And so you might now consider that even though the civil engineering system was working to destroy certain types of microbes, because it was allowing other microbes to thrive that had picked up resistance genes from the human microbiome, they are now disseminating those genes all over Lima. So this is where, again, these kind of bioprospecting methods to hunt for resistance genes comes into play to have potential public health impacts. Okay, so I'm going to leave the human microbiome and spend a little bit more time on a habitat that maybe a lot of people may not consider as a really key part of antibiotic-resistant exchange, and that is the soil, or the soil microbiome. So why look at the soil? Well, as it turns out, there's a long history, decades worth of really important work implicating the microbes in the soil, perhaps the most diverse ecosystem of microbes on the planet, in not just the production of antibiotics, but also in antibiotic resistance. And I'm only going to give you 3 examples of work by other people that really inspired our work. One was this beautiful story by Jerry Wright and colleagues, where they went into the Canadian Beringian permafrost, cored out samples that they could carbon date to be 30,000 years old, sequenced the DNA there, and then interrogated that DNA for whether they could find antibiotic resistance. And lo and behold, they found resistance to modern antibiotics, providing clear genetic evidence that resistance in environmental microbes vastly predates any human use of antibiotics. So kind of putting the nail in the coffin of saying resistance existed before we came up with antibiotics or discovered them. Another piece of evidence that's important is put forward by kind of the grandfather of the antibiotic resistance field, Julian Davies, who now more than 40 years ago recognized that almost all of these compounds that we call antibiotics are natural products of soil bacteria. Right, so all of those Nobel Prizes that were given for the discovery of antibiotics came from people hunting in the environment. This is the case with aminoglycosides as an example, where you found these extracts from the soil and realized, whoa, they're able to kill these particular pathogenic bacteria. And it's because of these small molecules. And so what Julian posited, and it's been supported since, is that these producers must be the original evolutionary progenitors of antibiotic resistance. And why is that? Because these antimicrobials target, as I said, key processes of bacterial life. So if they didn't have resistance at the same time as production, they would commit suicide. So it would not be a terribly interesting evolutionary process to figure this out without having resistance just in time. Since all of this happens, millions of billions of years ago, they have also given all of their neighbors the pressure to evolve resistance. Hence, soil is the original source of resistance. And then finally, to show that we as humans can actually have an impact in a bad way, this is evidence from European archival soils of about 70 years, where this group led by David Graham was able to measure the abundance of resistance genes and find that over the years of human use, their abundance has gone up. So based on all of this evidence, the one thing that you might speculate is that for this reason, every resistance gene, every resistance element that you find in a pathogen, a disease-causing microbe, must be the same as the one. They must be identical to things that you find in the clinic, in the soil. And bizarrely, that's not what we found, right? So when we started working on this and looking through databases, virtually all of the resistance genes that people had worked hard to discover in soil microbes were vastly different from the type of microbes that we see causing infection. So this seemed like a bit of a conundrum, and our hypothesis was maybe we've just been looking at the wrong slice of the pie. Now, one thing that Jerry Wright's group had done to really emphasize that the bugs in the soil are highly resistant was they went and they isolated, in this seminal paper from 2006, a whole group of microbes that they know are the producers of antibiotics. So all they did was culture them up, know that they produce of antibiotics, and then test it to see how resistant are these non-pathogenic bacteria to antimicrobials. And shockingly, on average, these bacteria were resistant to 7 or 8 different classes of antibiotics, even though they're not pathogens. So that's actually more resistant than most pathogens. So now, this should be used as evidence, probably, that these guys are the guys who are producing the resistance genes, right? But again, we stumbled across this problem where when people analyze the genes that these producers have, they look completely different from the genes that the pathogens have. So how do you reconcile that particular disparity between the phenotype of high resistance and the genotype of being very different? So one way in which you could reconcile that is to say, okay, I buy the argument that these guys are the original donators of resistance. But it happened so long ago, millions to billions of years ago, that it's not really relevant in terms of us treating clinical problems. So that's one hypothesis. The other hypothesis is maybe there's still another missing link. Maybe there are microbes in the soil that are contributing, we just still haven't found them out yet. And so this now jumps over to this crazy experiment that we did when we were in George's lab, where we said, okay, what about these weird bugs that are able to eat antibiotics, right? So you've got the producers, you've got the resistors, and now you've got the eaters. So in this slightly, again, ridiculous experiment, we tried to enrich microbes in the soil that could, again, not just resistant antibiotics, they were using them as their sole source of energy. It wasn't a terribly elegant experiment. It was just a lot of culturing. And when we were able to successfully identify all of these microbes that could eat antibiotics, we then tested their resistance. So now what I'm showing you in the bottom, in blue, is the same data from Jerry Wright, where his producer organisms, on average, resistant to about 7 or 8 antibiotics. Now you look at the eaters on the right-hand side. They're, on average, resistant to 17 or 18 antibiotics out of 17 or 8 antibiotics to which we could test. Basically, they don't care about the antibiotics. Actually, they do care about them because they're eating them, right? So we think now we had finally stumbled across that missing link, the potential that these eater organisms might be contributing to resistance in the clinic. Now, before I get to showing how we actually did that, I'm going to jump ahead 10 years from that original discovery, which is how long it took us to figure out how it was that these bizarre bugs were eating antibiotics. How are they surviving on the antibiotics the way that most bugs survive on glucose? And so this was work that was led by many people, but primarily by Terence Crofts, a recent postdoc in my lab, where he went to about 5 of these soil bacteria and used a battery of techniques to really pin down how the heck they were using those compounds which normally kill bacteria as their source of food, right? And you could almost brush past it if I don't point it out. One of these bacteria can eat penicillin as its sole source of carbon, but it can't use glucose. Doesn't like sugar, likes antibiotics, right? And so I'm not going to belabor, again, the molecular biology. All I'll say is that through a lot of different people's contributions, we were able to figure out that there are 3 key steps of how these bacteria eat antibiotics. And the important part is that the committing step is an antibiotic resistance gene. So the very, very first thing that these bugs need to do is still detoxify this toxic compound. So they break it open exactly the way the pathogens do, and then they digest it using a couple of different methods. And just to show that we really, truly understood it, one way in which to show that the genes that underlie this particular phenomenon are sufficient for this particular trait is you could transfer that trait to another bacterium that doesn't have this, right? So that's something else that we did, led by Terence, was we were able to take all of that inference he made from these soil microbes, transfer those genes into E. coli, one of these benign microbes in the lab at least, and he was able to convert E. coli into an eater of penicillin. Now, we're not doing this because we're crazy. Someone might ask, why the hell are you turning E. coli, which can, some versions make people sick? Why are you making it eat antibiotics? Well, in this case, our motivation was partly to prove that we really understood how it worked, but also because it might be used in the future for cleanup of antibiotics, right? Not with this basic work, but perhaps in the future where if you can have microbes that have these abilities to destroy these antibiotics, which can contaminate systems, like we had seen in the sewage systems, maybe this is a next generation approach to clean up, much like we clean up our sewage, using microbes to destroy these antibiotics before they go out into the wild. Okay, so now let's step back to say, right, so that showed that we know how to now, we can figure out how they eat the antibiotics, but what about this fundamental question? Are they the missing link between the soil and the clinic? And so to do that, we stepped back, and this was work done by Kevin Forsberg, a graduate in our genetics program, and Alejandro Reyes, who was a graduate student in Jeff Gordon's lab. So they applied that method many, many slides ago, where we go and discover new antibiotic resistance genes, and they applied it to these antibiotic-eating bugs and said, okay, using this method that doesn't require a priori knowledge of what resistance genes are, looking for cryptic resistance genes, can we discover the missing link? And sure enough, they did. In a single experiment, they now increased evidence by an order of magnitude of the number of genes in the soil that were exactly the same as resistance genes in pathogens that cause disease. And again, I won't talk about the specific details of the resistance genes in terms of what their names are, but the key features that certainly made us a little bit scared was the fact that between those 10 genes, they knock out 4 or 5 classes of antibiotics. We find them not only in those bugs, also in pathogens. And in that map that you see, any country that's shaded dark is a country that has deposited, or someone in that country has deposited a pathogen with a resistance gene that was identical to those genes we discovered in US soils. And to emphasize that this was particularly problematic, what you're seeing here is a comparison of all of those genes compared to genes that have been found in pathogens. And you'll see that there are many genes there, right? So across the bottom are the genes that come from the soil. Across the top are the genes that come from 4 or 5 important pathogens. And anywhere you see gray shading, that's identical, 100% DNA identity. And what we observe here is that not only do we find that they're the same single genes across these bugs, but actually they cluster together. So anything in red that you see is an antibiotic resistance gene. Anything in yellow is a gene involved in horizontal gene transfer. So they're the vehicles that move genes around. So what we're seeing here is evidence that bugs in the soil and bugs that cause disease can move large chunks of DNA between themselves that can knock out 3-4 classes of antibiotic resistance in horizontal gene transfer events that could happen on the order of minutes, right? So again, now, taking all of this together, reconciled with the data that was before, it suggests that we have discovered the missing link, and these are the bugs that we might want to focus on as we consider challenges to the clinic. Now, I will again preface all of this, I guess, by saying that we stumbled across this, right? This was not something we originally set out to do because, only someone insane would think about looking for antibiotic-eating bugs. But since we had this, and now we had this data in hand, we wanted to ask, how much of this is this the exception versus the rule? That is to say, because we had gone through this bizarre experiment of culturing our bugs that could eat antibiotics, which we discovered were these multi-drug resistant proteobacteria, now Kevin, who led that study, teamed up with Sanket Patel in the lab to ask, again, when we stumbled across those weird bugs, are they representative of all bugs in the soil, or are they sort of these weirdos, right? And basically, the short answer to that story is they are weirdos. And really, the only important part of this sort of complicated slide was we now collaborated with a couple of ecologists in Colorado, Rob Knight and Noah Fearer, got access to a much larger number of soils, about 20 soils, applied the same methods, discovered thousands of new resistance genes in the soil, but luckily, most of those resistance genes are not ones that we found in pathogens, right? So it appears really it's only that tiny weirdo minority there of proteobacteria that are the ones that are the missing link. The other thing we discovered is all of those genes we found from these bugs that are not the same as in pathogens, they don't have those same linkages with mobilization elements that cause those genes to hop around, right? So we know that in pathogens, for those genes to move between different bugs, they require those accessory elements to actually move the DNA back and forth. These guys in the soil didn't have them. So how do we put all of this together towards something that could be impactful for work in the future? We think what we've done is we've discovered, indeed, that the soil is rife with antibiotic resistance genes. probably reflecting this evolutionary pressure of antibiotics being produced in the soil. But fortunately, most of those genes are not at risk for being acquired by pathogens. But there are these very specific bugs, but now we know the identity, that we can focus on so that we can prevent them from traversing these particular boundaries. Okay. But as it turns out, there is still something interesting to learn from these particular bacteria that have, from the soil, that have these cryptic resistance genes. So one hypothesis that we came up with at the time is to say, could these be bellwether events? Could these genes that we have seen that haven't popped into pathogenic bacteria yet, could we stack rank them? Could we evaluate their risk across those thousands for the few that we think might emerge into the clinic in a few years? So why would you want to do that, right? Why would you want to know what is going to happen in the future? It's because if you know which ones are at great risk, those are the ones you can focus on for mitigation, the ones that you might try to destroy, the ones that you might try to diagnose. And this is the story I'm going to tell you about one such class of enzymes. And that's against the class of antibiotics called the tetracyclines. The tetracyclines are a very, very important class of antibiotics. They were discovered in the sort of late 40s, and they continue to be one of the major classes of antibiotics used in the clinic, in agriculture, and aquaculture. And the way they work is they gunk up a bacterium's ability to make proteins, right, by targeting this thing called the ribosome. So, big surprise, because they've been used against pathogens, pathogens have figured out how to be resistant to them. And the two main ways that they do that is they either pump the drug out, or they protect their ribosomes. What hadn't been observed very much, though, was a third mechanism of resistance, which a lot of bacteria use, and that is destroying the chemical itself, right? And that seems to be a pretty good idea if you're a bacterium in terms of wanting to be resistant. If you destroy the chemical, it can't hurt you, and it can't hurt your population. That's how, for instance, those beta-lactams, those penicillins, that's how they're destroyed by most pathogens. There are enzymes that cleave them and break them apart. So we found evidence of only one such instance. There was a non-pathogenic bacterium from human guts that had been shown to have this particular enzymatic activity. So we decided to say, we've got this treasure trove of newly discovered resistance genes in the soil. Could we find evidence that these guys might actually be lurking somewhere beneath the detection threshold? Motivating this was, again, as I mentioned, the fact that the tetracyclines are a unique class where not only are they old, but they've kind of gone through this renaissance of being modified and getting newer and newer versions of them, including, and this is something that's really important to keep in mind, the last 3 drugs that have been approved by the FDA in the US as antibiotics are all tetracyclines. 1 in August of 2018, and 2 in October of 2018. So a really important emerging class of antibiotics. One other thing to note is that those drugs that are coming to market have not been considering, obviously, a resistance mechanism that have not been seen in the clinic yet. So we want to make sure we can enable the protection of these particular compounds by seeing whether there, again, is this lurking danger against them. So to do that, Kevin, who had done the soil work, basically discovered this new class of enzymes across his soils. Again, I won't walk you through all of the data. All I'll say is that he was able to show that about 10 such enzymes existed that can destroy the tetracyclines and a whole bunch of soil bacteria. They were completely novel. They were not related, for instance, to the ones that have been seen in the soil, in the human gut before. And the only gene or enzyme that we could find in any database that they were similar to was a gene in Legionella. So Legionella causes Legionnaire's disease. It's a soil-derived pathogen. And sure enough, this was something lurking in the genome of Legionella that no one knew was a resistance gene that we were able to now show can cause Legionella to no longer respond to tetracycline therapy. So that's all well and good. What is the clinical relevance? So it turns out Drew Gasparini, who recently graduated from my lab, went through all of those selections that had been performed in my lab across many, many, many different habitats, from human fecal samples and soils and sewage samples from El Salvador and Peru, and started looking to see whether maybe more of these are there. In fact, through just a quick cursory survey, he found that there were 70 more such enzymes that existed just in our freezers, right? And in this case, good for our funding and really bad for humanity. Just in the time that we started working on this, those resistance genes started popping into pathogens, right? Exactly what we had speculated would occur. Really hoped that it wouldn't, but it did, right? So this is why we've now decided to take a very concerted effort to understand those enzymes, understand their vulnerabilities to hopefully destroy them. And so that's exactly what we've been able to do. turning back to something that we've learned from the penicillins. So one way in which the chemists have helped rescue the penicillins a little bit is to recognize that, yep, the penicillins, or the beta-lactams, are degraded by these particular professional enzymes called beta-lactamases. So if you can come up with chemicals that break the resistance, you can reactivate the antibiotic, right? So this is the idea of a beta-lactam, which is the antibiotic, and a beta-lactamase inhibitor combination, so breaking the resistance. So we said, hey, why not try that? Let's try to find compounds that kind of look similar to the tetracyclines that might be able to gunk up those tetracycline destructases. And again, this is where serendipity comes into play. You may not believe me, but the very first compound we tried had that property. And it happens to be a compound called anhydrotetracycline. The name is not all that important, but again, what we're able to show is that if we add anhydrotetracycline into the mix when these enzymes are working to degrade the tetracyclines, they don't work anymore, okay? And what you're seeing here is something that, that's a little bit more relevant to the clinic. There's a way in which to measure the resistance that is encoded in an organism by looking at how well it will grow in the presence of the antibiotic. So on your left-hand side, you're seeing what's called an e-test strip. This is basically a piece of paper that has an antibiotic across a gradient that you put on a lawn of growing bacteria. If the bacteria can't grow close to it, they're susceptible. They're being killed by the antibiotic. If they go really close to it, they're resistant. So you can see in the first panel, the bacteria are growing really, really close to it, right? So this is E. coli with one of these enzymes growing close to tetracycline. They don't care that tetracycline's there. We add this newly discovered inhibitor, and lo and behold, tetracycline can work again, right? And so we are now really heartened by this discovery, and through collaboration by a couple of other researchers at WashU, an organic chemist, Tim Vincevich, who trained with Chris Walsh here, and Neeraj Tolia, a structural biologist, we have now been able to take this initial sort of pre-discovery almost, make new synthetic analogs to that originally discovered inhibitor, and now we've been able to make even more potent inhibitors of these particular enzymes. And again, now that we were spending more time looking at more and more of these genes, what we're seeing here about 20 or so of the genes that we've been working on so far. The ones in red are the ones from the soil. The ones in blue are ones that now we've discovered live in the guts of healthy humans. One of the things that's, again, motivating us to sort of work fast on this is I already told you one of these enzymes from the soil we'd found in Legionella. Just last year, we discovered that one of these enzymes that destroys every single tetracycline we've thrown at these particular enzymes, including the compounds that were just approved by the FDA. They destroy those too. That gene or that enzyme we found in a cystic fibrosis patient sample in Pseudomonas, right? So that's really scary. But the good news is our hypothesis came true. By focusing 4 or 5 years early on trying to find those inhibitors, we now have inhibitors that can resensitize that Pseudomonas isolate to those latest generation tetracyclines. Okay, so I'm going to end my talk now in the next couple slides with the simple question, why are we not dead yet, right? With everything I've just shown you, why are we not dropping like flies every time some highly drug-resistant bacterium comes around? And certainly, the CDC has this almost Pokemon card deck of bad guys that tells you should really be scared, in terms of their hit points. You've got Acinetobacter and Carbapenems, resistant Enterobacteria, and Pseudomonas, these beautiful pictures of things that if they get in you, they're going to kill you, right? So what can we do about this? I showed you one strategy. Do we have anything in our arsenal to be able to fight back against these multi-drug resistant organisms? So it turns out there are some strategies outside of just coming up with new antibiotics that we might be able to use. And this is where we turn to some evolutionary biologists and systems biologists to think sort of out of the box a little bit, to figure out what are general strategies we might have to kind of flip the table on the drug-resistant organisms. And so one idea is called selection inversion. So what does that term actually mean? It means trying to come up with some kind of magic sauce that transiently allows the drug-susceptible version of the organism, the one that you can kill, to outcompete the drug-resistant version. Right? Why would you do that? Because as you're shown here, normally you've got the susceptible and the resistant guys mixed together. In this case, the yellow bugs are susceptible. the blue bugs are resistant. Normally what happens is you bring in the antibiotic. Sure enough, the susceptible ones by definition, the yellow ones get wiped out, the blue ones survive, and now you've got a drug-resistant population. Selection inversion, this magic sauce, is somehow to suppress the blue-resistant guys, to let the yellow ones take over, and then you kill them with the antibiotic. So how do you do that? There are a few strategies that have been proposed. One is drug cycling. So if you were able to find drugs that work in very different ways, what you might do is use drug A until you get a resistant population to drug A, and then much like crop rotation, you switch to drug B. Because those resistant mechanisms don't necessarily talk to each other, you can go back and forth, and kill off these differently resistant populations. This is only, unfortunately, going to work for a short period of time, because as I showed you in the soil case, sometimes multiple resistance genes can transfer at the same time, and then you've lost this particular ability, right? So another strategy is to recognize that drugs of different classes sometimes can combine together to be better than the trivial sum of their parts through a property called synergy, right? So what you're seeing here in this particular panel is normally you take a population that if you hit it with drug A, it doesn't care. Individually hit it with drug B, it doesn't care. But when you combine drug A and B together, there's something magical about that combination where they potentiate each other and they can synergistically, in combination, kill those bacteria. So that's strategy 2. And then strategy 3, which I think is actually the most elegant, is this concept called collateral sensitivity, right? So if you try to explain what that term is, imagine for a second that as a bacterium becomes resistant to one class of drugs, class A, the mechanism that it uses to become resistant opens up an Achilles heel. Something about what it's doing to become resistant to drug A opens up a vulnerability against drug B. That's the collateral sensitivity. Because of resistance 1, you become susceptible to drug 2, if you will. A St. Louis summer analogy for collateral sensitivity is, imagine for a second you want to go out into the St. Louis summer, and you don't want to be hit by mosquitoes or bitten by mosquitoes, and you also don't want to drop dead because of the heat. Now imagine that what you decided to do is to make sure that you don't want to get bitten by those mosquitoes. You put on a big moon suit and you walk out into the St. Louis summer. You've certainly protected yourself against that mosquito, but you're going to drop dead because that moon suit is going to suffocate you in terms of the heat. That's the kind of weak analogy for collateral sensitivity. So I'm just going to end by saying we were able to implement this against one of these scourge pathogens, methicillin-resistant Staph aureus, or MRSA. So MRSA is a deadly bug. It kills something like 11,000 Americans when it gets into the bloodstream. It's resistant to the important class of beta-lactams, these penicillins. And so we thought, could we use some of those features that I told you about before in combination to try to knock out MRSA? And so in this story, Pat Gonzalez, a graduate student in genomics in my lab, was able to find a combination of 3 beta-lactams, so in that same penicillin category, completely FDA-approved generic drugs that on their own are completely useless against MRSA. But somehow, in combination, they work together to overwhelm MRSA's defenses and are able to wipe it out, right? Through this process of synergy. But perhaps more importantly, they have this process or this property of collateral sensitivity. That is to say, if MRSA becomes resistant to any one of those drugs, it now becomes more susceptible to the other 2. And why is that important? Because if you imagine you're a bacterial population, you've got these 3 drugs around, you think you're clever and you become resistant to one, now you've got 2 other drugs still around that are much better at killing you, which locks them against resistance. And we tried very hard to make these guys resistant, we couldn't. And that was fortunate. And then finally, in collaboration with Maylin Chang and Shahryar Mobashri, researchers at University of Notre Dame, we tested to see whether this really interesting test tube result could actually translate into an animal model. So they happen to have an animal model of really severe bloodstream infections with MRSA, and we're able to fortunately show by working with them that this completely cleared that infection, this triple antibiotic combination, within 24 hours of treatment. And so we're pretty jazzed about this because, again, this is composed of 3 generic drugs, and so we have a lot of information about how they work. Fortunately, they're all co-delivered in the same way currently, and so we're hopeful that this might be one particular quiver in the arsenal against this deadly infection, but also sets up a paradigm of how we might be able to repurpose other existing drugs to come up with at least stopgap measures against infectious diseases. So with that, I'll end by not addressing this particular elephant in the room. And that is to say, everything I've said is focused kind of on human use of antibiotics. But in this country still, it's estimated that something like 80% of antimicrobials by weight are still used in the rearing of food animals. And I would argue, without getting into that debate just right now, that that's perhaps not a great use of this natural resource, right? And there are different ways in which to mitigate that, but I just want to say that this is something I've not discussed today, but I think is very germane to this particular discussion. With that, I'll also say that sometimes I look like this, but sometimes I also look very Missouri with my lab. I'd be remiss not to again say that really anything of importance that I might have said today is because of the wonderful people that I have the privilege of working with, both the people in my lab, alumni who've gone on to bigger and better things, people at WashU and around the world, and obviously the funders that are generous to support this work. And if there's one sort of PSA thing that you remember when you leave today, it's this: please don't use antimicrobials for viral infections. I'm happy to take any questions.
Thank you very much. That was great. I have a question about teaching. So part of the reason that we started this Undiscovered series was to talk about how we have to teach science and how we have to acknowledge these kind of unexpected findings. So I'm just curious, in your teaching now, genetics is a constantly evolving, relatively new subject. How do you incorporate this kind of thinking when you teach, especially undergraduates?
Yeah, that's a very good question. And I think there are already great measures in place, and we're doing this at WashU, where part of our undergraduate curriculum has a discovery element right off the bat. So there's this amazing thing called phage hunters, which a lot of universities around the country have committed to. And anyone interested, even if they're not declared biology majors, they have this sort of experience-based class where they'll still learn the fundamentals of microbiology and bioinformatics and sequencing, but they'll do it by going out into the field, for instance, harvesting soil, getting their own phages, these, the viruses that infect bacteria, going through the isolation process, going through the sequencing process and the annotation. So I think sometimes the best way to teach the sort of wonders of science is to actually perform the science yourself. Not everyone has the opportunity to immediately join a research lab. So I think engaging students very, very early on in discovery-based science, it's of course exceptionally important to teach them the value of hypotheses, but also to have that kind of exciting moment to say, no matter what you do in a phage hunters class, you're going to find something new. And then once you have something new, you can test a particular biological question. So that's one aspect that we find pretty useful. The other is my lab always hosts a pretty large cadre of high school students and undergraduates all through the year. Sometimes the people in my lab complain about this because we have a dozen students that come in additionally in the summer. But I try to convince people that you're doing this for multiple reasons. There's no other way to maintain the steady pipeline of students interested in science without allowing them to do the science themselves. And also, the only way to learn to be a mentor is to mentor. And so that's why I strongly encourage every single person in my lab, independent of what career stage they're at, to take on an undergraduate or a high school student to be able to see what it is like to teach. So I think those are two ways in which it could work.
Thank you again. That was fantastic. So the general mechanism of drug resistance, or the way the bacteria has become the superbug, is pretty well understood, thanks to you and others. So what is the practical solution, other than, to tell people not to take antibiotics and sort of sit out the cold, is something that most people are not going to, in practice, follow. So, in general, what would you suggest?
Yeah, there are a few things. Actually, I would say that education campaigns in countries that have tried them to pretty openly discuss when it is appropriate to take an antibiotic, Canada as an example, have been successful. It's just that the timelines of turning people's opinions around are not on the order of weeks or months. They could be years, right? And so I think reframing antibiotics as a natural resource, as something that's finite, and that's something that really requires a sort of conservation mentality, and then tie that to the fact that there is collateral risk that you're taking when you sort of willy-nilly take an antibiotic. I think a large part of being able to turn that table to sort of preserve those antibiotics when we need them is education. But when I say education, I just don't mean to the consumers of the antibiotics, but it's also to empower physicians, pediatricians, for instance, to say, here is why, ultimately you're still going to have to respond to the really aggressive parent who says, I don't really care, I want this drug for my kid, fine. But if you can spend maybe a few minutes to explain to the parent, here's why, here's a brochure, here's something that you should consider, here's the risk that your kid 5 years down the line because of this unwarranted antibiotic has a greater chance of something bad happening, I think that is a large part of change in practice, I think, for turning the tide there. I think the other also is sort of better reporting structures, right? So we, in clinical fields and biomedical fields, have to report every single antibiotic used. We still have no legislation in this country, and it's never been the case, where any agricultural use of antibiotics needs to actually be reported in quantitative scales. And I think that's a problem.
Thank you for your nice talk. I just wondered, so given the current technology of human manipulation of genes, especially gene editing, so convenient, widely used by humans, I just wonder this, does this in any way affect the antibiotic genes, evolution, speed of changes?
Yeah, so let me just restate your question so I understand it. If you're asking whether the ability to gene edit in a human genomic context has impact here, I don't know.
But all the other, like bacteria. And
right. So the same methods that people have heard in the media, right? These CRISPR systems that can go out and very precisely edit various aspects of any genome. There is a lot of promise to use methods of that type to perhaps go after the resistance genes themselves in the bacteria. So one way in which to counteract the emergence of resistance might be directed targeting of those resistance genes and to blow them up. And there's been some really, really promising work from a lot of researchers here in the Boston area to show, at least in proof of concept, that that can happen. And the reason that those might be really elegant methods is because the reason resistance has a really high potential of arising is because you're challenging the life of the bacterial population, right? It's a life or death decision, so there's immense selection to be able to become resistant. In principle, if you were to destroy the resistance gene, then you might be able to get non-resistant versions of those bugs to survive, and the selection pressure might not be as high. So yes, absolutely. I think those gene editing methods might be very useful as part of the arsenal in the future to help mitigate this problem.
Thanks.
Hi, thank you for the talk. This is very interesting. I have a bit of a technical question about the strategy that you use for discovering potential new antibiotic resistance mechanisms by transforming them into E. coli. Some genes would definitely be and have been discovered in this way, but there definitely could be genes that even if you put them in E. coli, they're not compatible with the machinery of E. coli, they wouldn't actually protect it from the antibiotics, but they would some other bacteria. Do you have any way of estimating the proportion of how much you're missing out versus how much you're finding?
Yeah, that's an excellent question. And so, even to expand on that, E. coli is in this group of organisms called gram-negative bacteria. And there are a whole class of really important pathogens that are gram-positives. And the reason this detail matters is those drugs don't work against the gram-negatives at all. So E. coli is intrinsically resistant to those antibiotics. We would never discover any resistance genes against those important antibiotics like vancomycin and linezolid. So one way of which to address that problem is take exactly the scheme I showed you and swap out E. coli for another host bacterium that has the properties you care about and do the same type of screen, right? But then the direct answer to your question of the estimate of how much we might be missing is, again, we think that is biased towards particular bacteria. So the way we've estimated that in the past is we've done that same experiment, but with the DNA material that goes in from defined bacteria of particular types, for which we already know what the resistance genes might be, and then see which ones come through. And again, what we see is that there are two layers of bias. One layer of bias obviously is against any of the gram-positive bugs. Those mechanisms just don't come through. And then we also have a little bit of bias towards, again, the kind of GC content and codon bias, the type of things that appear to be more distant from E. coli. But the good news with E. coli, and I think we got kind of dumb lucky with this in terms of it being this model organism, it is pretty promiscuous, right? So it's transcriptional and translational machinery, the ability of DNA into RNA, RNA into protein, is sufficiently promiscuous that a lot more genes come through than if you were to, say, use just by arbitrary nature some other microbe. So.
Thank you for your talk. Could you explain more about the difference between drug cycling and the collateral susceptibility? Because it seemed like, from what I understood, the collateral susceptibility, one drug resistance opens susceptibility to another. So wouldn't you just be cycling drug B or C? Or?
Yeah, great question. I should have said that none of those are mutually exclusive, right? So in fact, people have suggested that when you do drug cycling, you should use collaterally sensitive drugs, right? It's just that you could also then use collateral sensitivity in combination. So you can combine any one of those different methods to be able to come up with novel ways in which to repurpose drugs. So drug cycling is just the very general concept of using multiple drugs one after the other, like crop rotations. Whether you use collaterally sensitive versions or not, it might still work. If you happen to use collaterally sensitive versions, it might work a little bit better, right? But you could also use collateral sensitivity without any cycling, basically by using it in combination. So that's really the clarification.
Thank you. It was a great talk. I had a question, however, about the inhibitors of the mechanism that inactivate the tetracyclines. Because by analogy with the beta-lactamase inhibitors, all you do is you select for mutations in the resistance gene. So presumably that will also happen.
Yeah, that's a great question. And probably the most honest answer to your question is eventually the bacteria will win with any single drug, right? They're just evolutionarily smarter than we are. So how do we try to extend the lifetime of a new compound? So one thing we've done, and I didn't show the data here, is we focus on a structural approach. So we're solving the structures of all of those enzymes with both substrates and inhibitors bound, and trying to rationalize why the inhibitors are working. And by rationalizing that, then we can use more sophisticated structural methods to say, hey, maybe we can lock these particular positions in these inhibitors through synthetic design such that it's less easy for those particular enzymes to mutate. So in a way, and this is something that's a great part of biology, is you actually, anytime you think you found something good, you try to break it, right? And once you break it, you understand what the risk of the later on mitigation might be. So we're doing two things. We're adaptively evolving those enzymes to be even better, right? Not because we want to release them into the environment, because we want to know what the risk might be. Trying in an amino acid specific way in the structure to understand how mutable they might be. And then designing our inhibitors through chemical synthesis to allow that not to happen. Again, that's never going to be the end of the story, but it'll give us a longer therapeutic window, we think.
Thanks very much. I'm just rather sad that my era as a working scientist happened before all of these modern techniques were available. But anyway, it's great that you're using them.
Thank you.
Okay, so thank you so much for the presentation. I really appreciated it. My concern during the whole presentation was that the antibiotics, while fighting diseases, they actually fight even our body. And we cannot even go back to the evolution, we're just moving forward. And my concern are two, though. The first concern is that with the evolution, even, for bacteria and for all the diseases, evolution is the same for humans. Not practically or expressively, but it exists. And when antibiotics go to our body, they fight our body, as well as they fight bacteria. And I'm concerned that, do you believe that we should keep the focus on antibiotics, or we should try a way to find something more biotic into our body, that instead of fighting our body, support our body, and together with the body, fight the disease?
No, I think that's a fantastic point, right? That we've been kind of stuck in this warfare mentality with microbes because that worked, right? Back when the antibiotics were found. And it worked really well. We're happy that they worked while they did, but they're going to have collateral impacts. Now, it's true that there are some toxicities involved with antibiotics, but for the most part, the doses that we use a lot of antibiotics are relatively safe. But we still have this issue that we are taking these chemicals in that are going to have all of these collateral impacts, at least on the other good bacteria. So the neat part of microbiome science, right, all of this focus on microbial ecosystems and communities is beginning to let us learn from that in terms of saying, how is it that microbes naturally interact? they interact in communities, they work with each other, sometimes benignly, sometimes in conflict, but it's still at a community level. So can we learn from those principles to come up with new therapeutics of the future that are not purely these offense molecules? And there's amazing work being done right now in basic science and also in industry to do things like either design a probiotics, specific microbes that can be engineered to deliver particular products or to modulate other microbes, and also other types of chemicals that are not offense molecules, but they might allow one particular microbe to grow a little bit better than another type of microbe, so they're potentiators of maybe good bugs that can crowd out bad bugs. So I think you're absolutely right. The therapeutics of the future will not simply be offense molecules of the types we've had. They will include them to some degree, because no matter what I say about collateral damage and all of that stuff, if you're infected with the plague bacterium, right, you shouldn't worry about wringing your hands about whether you should take antibiotics or not, right? Yersinia pestis is not going to respond to some touchy-feely FMT, for instance, right? You're going to have to kill that bug. But for the future, for other types of infections that require a little bit more moderation, I think there are incredible new avenues. And I think, for instance, one of the really cool features of the microbiome that I'm really attracted to as a future therapeutic is its direct interface with the human immune system, right? So we can modulate microbes, even at a community perspective, be able to very carefully tickle the immune system to then trigger it at the appropriate time to be neither underreactive nor overreactive and go after specific pathogens may be an incredible therapeutic avenue for the future. And I didn't have time to talk about this, but we have a big arm in our lab of our engineers who are working on probiotic engineering, both bacterial probiotics as well as fungal probiotics, to do exactly the type of things I just talked about. Deliver enzymes that are specific for metabolic disorders, but also to, for instance, display very specific tags on their outside, on their surfaces, specific to bad bugs, to be able to take them out of the ecosystem.
My only concern was that we were so focused on destroying the problem, and we were not talking much about safety, and that was my concern. So thank you so much.
Yeah, pleasure.
Hi. Regarding what you said about the use of antibiotics in agriculture, now obviously that's in large part due to the fact that it just makes the process of animal husbandry much easier and more efficient. However, there is a lot of evidence showing that there is a lot of deleterious effects emerging because we are, using antibiotics for that, and it's, hurting our ability to fight disease in humans. So do you see any potential ways that we could reduce the use of antibiotics in a way that makes sense economically for these companies, or do you think legislation is going to be the major mechanism?
No, I think that's a brilliant question also, because the agricultural industry in the US is an exceptionally important part of our economy, right? And I think just wantonly banning the use of antimicrobials is not going to work. In fact, we've seen experiments in this ilk in Europe, for instance, where we'll ban the so-called non-therapeutic use of antibiotics, wash our hands of it and think everything is good. And then what will happen is the industry is incentivized to change the concentrations of what's called a therapeutic use. Then the drug use goes back up. So I think working with the industry and coming up with solutions to mitigate and come up with alternatives, actually I think the best alternatives right now are back to microbiome science. Right? Why are these antimicrobials working to make these animals that much better in terms of their productivity? It probably has something to do eventually with something on the host or something on the microbiome, right? So there's some really exciting work already going on, certainly in academia, but also in a few small biotech companies, where they're trying to understand whether you can deliver probiotics of particular features in terms of, again, compounds that are not antimicrobial, but might give boosts to particular microbes that allow the animal to be a little bit more productive. And now you're giving your farmers an option that allows them to still have meat that costs less, and so it's more productive, but then saves the antimicrobials for us. So I'm not saying that's a panacea in terms of a solution, but it is one way in which we can use all of this incredible work that's going on microbial ecosystems, understand the mechanism why the antibiotics work in the first place, and then replace them with a microbial solution.